Okay, so go to the next slide. It should say modeling ecosystem. So I won't spend much time on this other than to say that we're ultimately interested in building models of these biological systems, and there's many kinds of models. There may be models that are very mechanistic, that explain, say, how an individual protein mechanics might work. There are models that are more functional, that are not necessarily detailed about the mechanism, but they relate inputs and outputs. There are also models that are quite general, that are higher level abstractions that exist in ecology or in biology. And ultimately what we're interested in is using all of these models to help advance our theoretical understanding. And this is in some way how building this infrastructure that can be used by many people and can tie to high performance computing, where we can explore these models, will lead to some theoretical advances in biology and hopefully conceptual advances in our understanding of biological systems. Next slide. So this says, what do we need to advance theory? So this is essentially the outline for the rest of the talk. And it's more or less the 6 things that we're trying to do. So the first bullet here is, in order to build an environment to really explore modeling and theory in biology and to make systems that can be predictive in biology, we have to first collect as much of the relevant data into one place as possible. So that's step 1, collect observations, hypotheses, experimental results, DNA sequencing data, phenotyping data, expression data, everything, capture all the relevant data, integrate it together. So the second bullet here is we have to sort of organize that data, we have to classify it, and we have to integrate it. What we mean by integration here is tie different parts of the data together. For example, if we have a genome sequence of a particular organism, we have individual genes, we want to tie those genes to the protein, to what we understand about the proteins that they code for, and what the functions of those proteins might be, how those proteins interact with each other, and so on. So you can imagine this data as a set of links that connect it all together. That's the integration component, and that in itself is a very difficult challenge. Then we want to devise easy means to search, query, analyze, and compare this data, and to constantly update it. One of the ideas that's really powerful in computational biology and bioinformatics is the idea of annotation, which means to add notes or information to the data as we learn more about underlying science that data is connected to. And that happens over time by many, many people. So often the systems that are integrating this data have an annotation component to it, where people are allowed to update and attach notes. Then we want to build, and we've done all of this so far. The fourth step here is to generate the tools that allows us to model, to build models from this data, and to use this data to design experiments and to create conjectures or predictions. And in some cases, these conjectures are sort of functional, like we predict that this gene that we're not sure what it does, we predict that it could do function X, and then we encourage somebody to go in the lab and test it to do function X. In some cases, we're building integrated models of, say, an entire metabolic network, and we're going to make predictions of what the functional capabilities of the network are, and again, make predictions of how that network would behave, and to encourage experimental groups to validate those. In addition to creating individual models and being able to simulate systems based on those models, we often want to be able to compare models and visualize models, and then take new data from experiments and update these models. So models become a first-class entity eventually in this system. Once you have a lot of models and you're playing with them and you're understanding higher level principles that are true across many models, then you want to capture these new principles or generalizations or abstractions from collections of models, and that's how we push towards theory. And then finally, layered on all of the infrastructure, we need to have the ability to allow people to collaborate, because this is not a kind of science that people do individually. It's very much a group science, and at every stage of the process, there's a need to interact with lots of people. Next slide, please. So just to give you a sense of how big these these systems are we're dealing with. Back in 2001, when we started working on this kind of a project, there were about 300 databases, not 300 items of data, but 300 separate databases that contain many different kinds of data. When I updated this list last year, there were over 1,000 databases. These numbers here are from 2001. What this means is that not only is the volume of data increasing, which we sort of expect as sequencing and other technologies become cheaper, but the number of people in the community that are taking on the job of curating data is increasing. So we've had a factor of 3 increase in the number of groups publishing data in about 10 years. Next slide. This is just a snapshot of one effort many years ago to integrate data, just to giving you a sense of how the databases are connected. These are not individual, this is not just within one database, this is actually the linkages between databases. What this means is that if you're building an integrated environment, you're really having to solve the problem of how do I reconcile data across many databases, and that's at the heart of building integrated biological databases. Next slide. I talked about the annotation. Of course, ultimately, I mean, annotation comes from the original Latin meaning to add notes to, or to note and mark. Came from the 5th century, probably, when monks were transcribing manuscripts and kept, adding notes to it. We take this idea and mean to add notes, add insight to biological functions, or biological data. Next slide, please. In a nutshell, that list I showed you of starting with the data and moving towards models, it can be represented sort of as a pipeline. And this is a pipeline, a simple high-level view of a pipeline that we've built over the last 10 years that allows us to go from complete genomes on the upper left through a series of steps to call the genes on those genomes, recognize features, categorize the genes that we find, assign functions to them, create mappings between those genes to reactions, and then build mathematical objects that represent the collections of reactions, ultimately on the upper left here to the point where we can run a model and make predictions. In this case, we're making what's called flux balance models that predict the flow of energy and material through the cell, and using those models you can make a class of phenotype predictions, like the type of culture media that's needed to grow an organism, or what an organism might, the growth rate of an organism under different conditions, or how an organism might shift from growing on one carbon source to another, and so forth. And so in some sense, we have a start on this generalized problem of how do we predict the phenotype, that is what an organism can do, from the organism's genome. And of course, in the process of building this pipeline, there's many auxiliary kinds of computational tasks you might do about studying, doing comparative analysis between many different organisms, building phylogenetic trees, studying horizontal gene transfer, and so on. And I won't touch on, I might touch a few of those. Next slide.